Hello everyone, my name is Troy, and today I'm going to take you through prophylaxis of hydroxychloroquine on COVID-19. Just to put a disclaimer, I'm not a medic by profession, and my suggestion is don't accept what you hear, don't neglect what you hear, and please don't reject what you hear. But first, let's understand who recommended the use of hydroxychloroquine on COVID-19. About 2 months ago, South Korea COVID-19 Central Clinical Task Force recommended some interesting treatment principles on COVID-19. They recommended the use of Kaletra, which is a combination of lopinavir and ritonavir on treatment of SARS-CoV-19. But why is chloroquine efficient in treatment of COVID-19? To understand that, let's review a little bit of molecular biology. So, this is the cell. Inside the cell, we have the nucleus. This is the nucleus. Inside the nucleus, we have DNA. And in a process called transcription, DNA is transcribed to create RNA. This is RNA. And inside the cell, you have the cytoplasm. So what happens is the RNA is released into the cytoplasm of the cell, and there it's modified, whereby there is a poly A tail added onto it and a 5-cap prime added onto it. So let me just give you a diagram of how the RNA has been modified inside the cytoplasm. So on one end, we have a poly A tail. And on this other end, we have a 5' cap added onto the RNA. So that's the RNA. Once that has been done, it signals to the ribosome. So this is the ribosome, that the RNA is ready for creation of a protein. So in a process called translation, the ribosome moves from this side to the poly A tail side, creating a protein. Now, what happens when a virus attacks a cell? So I'm going to give you a diagram of a virus and a cell and what happens. So this has COVID-19, and it has numerous proteins. And inside the virus, we have RNA. Now, this is our body cell. Our body cell has a receptor. This is called S receptor. S receptor 2. Now what happens is the virus uses this protein. This is called S protein. Or spike protein. spike protein. It uses the spike protein to attach itself to the S receptor 2. So once it has attached itself to the S receptor 2, the cell allows the virus into the cytoplasm. So this is the virus and this is the cell. So the virus, remember there was an RNA inside the virus. It releases its RNA into the cytoplasm of the cell. So once that has been done, replication of the RNA begins. And that's the replication of the viral RNA. So this is the viral RNA. It also has a poly A tail. And on this other side, it has a 5 cap. So that's the RNA, the viral RNA. So the ribosome translates this piece of RNA, moving this side, to create a protein. Now this protein is called RDRP. RDRP stands for RNA-dependent RNA polymerase. Now, once this protein has been created, the protein moves to this side. It moves to this side, creating a negative strand of RNA. So that's a negative strand of RNA, whereby the 5 cap is on this side and the poly A tail is on this side. Therefore, creating a protein. And this protein, the process continues, again walks to this other side, creating that piece of RNA, whereby there is a poly A tail on this side and a 5 cap on the other side. You realize we have ended up with the original viral genome of the RNA strands. Interestingly, this RDRP, at times it stops here, at times it stops here, creating shorter pieces of RNA. So on the first one, it stops there, creating that piece of RNA. On the second one, it might stop there, creating that piece of RNA. Now, these pieces of RNA are called subgenomic RNAs. And again, the ribosome thinks they have to translate these pieces of RNAs to create protein. So these pieces of RNAs are translated and we end up with pieces of proteins, the proteins that we saw on the virus. And now once these proteins have been created, they regroup together with the RNAs and they create or form a new virus, ready to attack a new cell, leaving the old cell dead. So that's what actually the virus does to our cell, leaving the cell dead. And I had a link here, and if you follow this link, it will actually tell you what the South Korea COVID-19 Central Clinical Task Force find out about the use of chloroquine on COVID-19. So it was realized that zinc could actually inhibit the replication of the RNA cells. So this is a molecule called zinc, and this is our cell. So the study shows that zinc on itself, because it's positive charged, the cell membrane is not charged. So it's not easy for zinc to penetrate a cell wall that is not charged. So they added a molecule called PT. They added a molecule called PT, and PT is an ionophore. What is an ionophore? Ionophore is like a means of transport through the cell membrane for zinc to get through to the cell. So PT was added to zinc to act as a mechanism or to make the cell membrane permeable for zinc to get through. And that study was done in vitro. In vitro, I mean in a laboratory, not inside the body. Inside the body, it's called in vivo. But in a test tube or in a petri dish, it's called in vitro. So this study was done in vitro. So what is an ionophore? An ionophore, as I said, is a means of transport. And that's where hydroxychloroquine comes in. Hydroxychloroquine in itself is an ionophore. So hydroxychloroquine is not treatment or is not a cure for COVID-19. Hydroxychloroquine has been used in this as an ionophore. So it is zinc that actually inhibits the replication of the RNAs in our body cells. Now you understand why hydroxychloroquine has been used in treatment of COVID-19.